Hello everyone. Today we are going to talk about CRISPR. But before we indulge ourselves into knowing how CRISPR works and its application in our lives, let us first understand what is CRISPR. CRISPR is short for CRISPR-Cas9, and the word CRISPR is actually an acronym, and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. So CRISPR is a segment of DNA containing short repetitions of base sequences involved in the defense mechanisms of prokaryotic organisms to viruses. So what does CRISPR do? CRISPR edit parts of a genome by either removing, altering, or adding sections in a DNA sequence, thus modifying the gene function. Now, Cas9 is an enzyme that acts like a pair of molecular scissors, or basically it cleaves the DNA. Here is an example of CRISPR-Cas9 gene editing. So starting from left to right, a portion of the DNA structure may be disrupted. If a single cut is made, a process called non-homologous end joining can result in the addition or deletion of base pairs, disrupting the original DNA sequence and causing uh, gene inactivation. Another one is deleting a sector of the DNA. A larger fragment of DNA can be deleted by using two guide RNAs that target separate sites. After cleavage at each site, non-homologous end joining unites the separate ends, deleting the intervening sequence. Lastly, it can correct or insert in between the DNA structure. Adding a DNA template alongside the CRISPR-Cas9 machinery allows the cell to correct a gene or even insert a new gene using a process called homology-directed repair. So why is CRISPR a groundbreaking discovery? So according to the scientific community, it is faster and cheaper. It is one of the cheapest techniques in genetic engineering and allows for the introduction or removal of more than one gene at a time. CRISPR is also more accurate, more efficient, and is not species specific. CRISPR isn't species specific, which means that it could be used on organisms previously resistant to genetic engineering. Also note that these are all in comparison to other gene editing techniques. Now, where did the idea of CRISPR come from? According to Vidyasagar, and I'm quoting now, CRISPR technology was adapted from the natural defense mechanisms of bacteria and archaea. These are single-cell organisms. These organisms use CRISPR-derived RNA and various Cas proteins, including Cas9, to foil attacks by viruses and other foreign bodies. They do so primarily by chopping up and destroying the DNA of a foreign invader. When these components are transferred into other, more complex organisms, it allows for the manipulation of genes. When was CRISPR discovered? So I made a short timeline of how CRISPR originated and where we are now with this technology. So starting in 1987, Japanese researchers found that there was a pattern of DNA sequences in a gene from E. coli. The gene has 5 short repeating segments of DNA separated by short non-repeating spacer DNA sequences. Each of the spacer sequences had their own unique sequence composed of 32 bases. Now moving forward to 2002, the term CRISPR was coined by a group of Dutch scientists, and on the same year, a second set of sequences was discovered, in which they are called Cas genes. By 2007, there was extensive knowledge about CRISPR-Cas9, and researchers started to conduct experiments. One of the experiments from a website stated that a team infected a milk fermenting microbe with 2 virus strains. Many of these bacteria were killed by the virus, but some survived and went on to produce offspring, also resistant to the viruses. On further investigation, it appeared that the microbes were inserting DNA fragments from the viruses into their spacer sequences, and that they lost resistance whenever the new spacer sequences were cut out. In 2008, Eugene Konin and his colleagues stated that whenever bacteria confront an invader, or let's say virus, they copy and incorporate its DNA segments into their genome as spacers between the short DNA repeats in CRISPR. The segments in the spacers provide a template for the bacteria's RNA molecules to recognize any future DNA of an incoming virus and help guide the Cas9 enzyme to cut it up so as to disable the virus. On 2012 to 2013, there was the first patent application submitted for CRISPR-Cas9 technology and publication of the new gene modification method. And from 2013 onwards, this gene editing method is used in different plant genomes and to modify human embryos. Fun trivia: on 2018, the first gene-edited babies was announced by Chinese scientists. However, on 2019, World Health Organization called on countries to ban experiments that would lead to more gene-edited babies. And on the same year, Chinese scientists were convicted for using CRISPR-Cas9 on human babies. Lastly, on the 7th of October 2020, Nobel Prize in Chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna for the development of a method for genome editing.